quantum chemistry at the National Institute of Applied Sciences in Lyon. He then obtained his title of Docteur-Ingénieur for his accomplishing theoretical conformational studies on polar amino acid residues, and afterwards his PhD degree for accomplishing theoretical conformational studies on nucleic acids of their constituents and on the related molecular systems. Oh, okay, so I don't have the year here, but I think this was a polite a polite move. I don't know if you agree with me. Well, he did both of these courses at the University of Paris IV, and then he held a researcher position at the Laboratoire de Biochimie Théorique at the Institut de Biologie Physico-Chimique in Paris, and at the Centre National de la Recherche Scientifique, aka CNRS. He was also a postdoctoral fellow at the Harvard in the laboratory of the Nobel laureate Martin Karplus. Professor Perahia was head of the theoretical simulation team at the Institut de Biologie Physique-Chimique Moléculaire et Cellulaire at Orsay for over 30 years. He was also research director at CNRS in 1991 and research director at the École Normale Supérieure de Cachin in 2011, where he remains as a research director emeritus since 2013. Professor David focuses on the study of the relationship between structure, dynamics, and function of proteins, and is actively involved in the development of new methods enabling the study of large conformational changes in proteins, having brought important contributions on different aspects of normal mode calculations on proteins. So Professor David, or should I say Davi, thank you for joining us, and yes.
It's too much honor, but I don't know if I deserve more. And, but thank you also for inviting me here and Paulo Beach. And after the COVID, it was 3 years, then is starting visiting Brazil. I'm very happy to come here. And then it's like a virus, no more than I catch it when I was with I can't take it out, okay. When I was in Harvard with Martin Karplus. And then we were developing all this with molecular dynamics. And it's like expanding virus like COVID, which is becoming very important field now. But it wasn't at the start. And then I prepared this mostly for the students to have an idea what is normal mode. And then it will be a little bit more technical in the beginning, but not so much, to give you the idea. And then I will present you some results, things like that. Okay. You know molecular dynamics, you are doing all molecular dynamics here. When normal modes, I don't know if you have done any normal modes. And then you know that molecular dynamics is just resolving the Newton equations. But normal modes also resolve the Newton equation. And you know that the protein, for example, macromolecule has many, many minima. You are exploring all these minima during the dynamics. And then you need a physical force field for that. Maybe you have also, learn this physical force field. And then you are doing numerical simulation. You are solving the Newton equation numerically, not analytically. But in normal modes, we do some approximation that in order to take, we have only one minimum here, which corresponds to a given minimum, for example, here. And then we have this paraboloid. Then we solve the Newton equation in this simple simple potential surface, which you can solve analytically. You don't need numerical simulation. Then it's very, very fast. Okay? And then you have a quadratic approximation of your potential energy function. And then if you have the potential function, you do a Taylor expansion, and you take only the quadratic terms of the, I don't know, you must know the Taylor expansion, but for the small displacement, then you can have all these quadratic terms, and then you forget this. If you want to forget this term, you have to be in the minimum, which in the minimum, the gradient of the potential is 0. You don't have any gradient. Then you can get rid of this by minimizing the energy. And then you can get rid of all the higher terms, which if you have small displacement, you don't need the third order term, fourth order terms, etc. And then if you use only these terms in the Newton equation, your Newton equation become another type of equation here. I will not present you the demonstration. Here is the mass. R is the displacement vector. You know, every atom displaces in one direction. Is the displacement vector. R dot dot here is the second derivatives of the position, which is the acceleration. If you have one dot, is the velocity. And if you derive twice with respect to the time, is the acceleration. Then you recognize here that m times acceleration correspond to the Newton equation. But we have another term, HR, which is the Hessian matrix. This Hessian matrix is the matrix where each element is the second derivative of the potential with respect to the coordinates. Then if you have n atoms, you'll have 3n lines and 3n columns. Then it's very simple to have the second derivative of the potential, because potential is very classical. Potential is very simple to derive and take the second derivatives of that. And then you have this matrix, which we call the Hessian matrix. And then here, this equation is a differential equation, is the second order differential equation, which the solution of a differential equation is an oscillation type. And here you have, for a given atom, the variation with respect to the minimum structure is a sum of oscillations. It's only vibrations. Then you have the frequencies, angular frequencies, omega k. We have the amplitude. We have some dephasing here. And then what we are looking on the normal mode is just to have preferential directions where you have only one oscillation, not a mute. Then you have the normal mode coordinate, what we define. In order to have these directions, privileged direction, then you have, I will not do the demonstration here, but if you take the eigenvectors or eigenvalues of this matrix, you know what is the eigenvectors, eigenvalues, then every eigenvector corresponds to a given collective variable vector, and the eigenvalues correspond to the frequencies, the square of the frequencies. And then you know that in matrix, if you have then the number, you have the same number of eigenvalues or eigenvectors as the number of lines. Then you have 3n normal modes. Okay, one normal mode is constituted by an eigenvector and eigenvalue. Okay, then you have 3n of them. You have a lot of quantities of modes. For example, for 1,000, for a protein of 1,000 atoms, you have 3,000 normal modes, each one having some frequency. And the most interesting ones are the very lowest frequencies, which correspond to very collective motions of the protein, which makes this method very interesting. Okay, then some facts. The movements in quadratic force field, as I showed you, are vibrational. Okay, solution of a differential equation of second order. Then overall motion is a sum of vibrations. Okay, I showed you this. And all the motions are independent to each other. In one normal mode, the motion doesn't know what is the other motion. They are completely decoupled. They are independent. They don't interact, these motions. And fourth fact is unidirectional. That is just going direction. There is some directionality. There is not curved motions. Okay. And then last fact is these small amplitude motions. Okay, very, very small. So tiny amplitude for a protein that even in your display, you don't see the motion. It's very tiny, okay? And then what is interesting here is that the lowest frequency modes, even that the motion are very tiny, correspond to the beginning of the very low valleys, energy valleys, okay, which you can go farther away, but you don't have the right to go farther away because you are limited to small vibrations. Then if you want to go very farther away, then you have to consider the whole potential, because here we took only the quadratic terms. Okay. Then other facts. Then if you do large displacement beyond the quadratic approximation, which requires a real force field, then you have what you have some couplings between the normal modes. If you have computed the normal mode, these normal modes will not be coupled, because if you move in one direction, this will affect the motion of the other motion of the normal mode. They are not uncoupled. And even that you can have direction, even the direction could be changed. If you go farther away, this direction will not be completely the same. You can have also changes in the spectrum, slight variation of the spectrum of the eigenvalues, small variation. Then the general picture is that normally, if you do a molecular dynamics, you have a combination of vibrational plus diffusive motion. What I mean by that, if you are, for example, this is your energy surface, then vibrationally, if you are in a one minimum potential well, you will be vibrating there. But if your temperature is quite elevated, then you can go over the barrier, and then you will be exploring all the energy surface. Then when you explore the energy surface, when you are going away, is which I call the diffusive motion. Then you have two types of motion, vibrational and diffusive. But when it diffuses, it vibrates too, okay? Then experimentally, this is observed with neutron experiments up to 200 Kelvin. This is the harmonic approximation, which this fluctuation, atomic fluctuation, is linear with respect to the temperature. But after 200 Kelvin, the fluctuation becomes very large, which is the diffusive region, because we have gone over the barriers. Then this experimentally been observed. Then what are we looking? Okay, as I told you, the lowest frequency direction corresponds to low energy value, the direction. If the low energy value is, I know this direction, which is very low energy, but it's for tiny, I'm a very tiny movement. Then if I have to go farther away, I have to push, give some kinetic energy, go beyond. And well, I don't know if these low energy values, that the low energy, this low frequencies shows this low energy value. I don't know if it is a general characteristics of any, I don't mean of a protein, any object, but maybe it indicates a low energy value, but is it specific to the proteins? But I think you have both of them. I think that even to any object, you have very low energy value, and the biology may be selected these motions, these directions of motions, in order that it can go even beyond what biology requires. And it's just maybe a selection of this. Okay. Then how do we do? Okay, another interesting thing is that we have all these low energy values. If I am in somewhere, then I have the Hessian matrix that I have diagonalized. I have all the direction of the low energy value. But if you do dynamics, you might not have all of them. You can do a PCA with the principal component, but it depends on your simulation time. And then what you do in PCA, you have the correlation matrix of displacement, and then you can give this motion. But the interest of the normal modes is that you have all of them. And then a good strategy to go beyond this tiny motion is to combine normal mode and molecular dynamic simulation together. Then you'll be exploring. This is a very dedicated name. We call this hybrid simulations. This is almost a new term now, which is being recognized like that. And then we worked a lot on this hybrid simulation. I think we were the first groups having done with Mauricio Costa, it's here, that we try to combine NM and MD, and also Paulo Batista is here. And okay, then we developed a technique which we called MDNM with molecular dynamics with excited normal mode, or A with normal mode in Brazilian. And then what we do is just we compute the normal modes of a given structure. And then this Q1, then we have the normal mode space. Normal mode space is constituted by the vectors, eigenvectors, as I told you. Here I put 3 normal modes, but you can have 4 normal modes, 5. But it's more simple to present a space of 3 normal mode space. And then for a given structure, we compute the normal mode. We have, I told you, 3n normal modes, but we have the lowest one. And then if, for example, you take the 3 normal modes, then we can combine them and then get a Q, which is a combination, for example, of 3 normal modes here. And then we can create a combination just to have an isotropic distribution of this vector in the space, okay? Then we can push the system by putting some velocity, push the system along the, if I am here, I push myself with velocity and I advance. But when you advance, you lose your kinetic energy, then you have to push again and go again and go again. And then you can go very, very far, like along a given direction. And then you can do for all the vectors separately, for example, and then you can fill the whole space with structures which correspond to very large scale structures, which we showed that is very difficult to get with dynamics. And then when you push the system with velocity, we have also excitation time, or you can say or relaxation time. When you put velocity, then this velocity dissipates, then you have to push again, and you have many times like that, and you can excite 10 times, 20 times to push very, very far. And then you feel this, and then you can analyze all the points, and you can do many, many things. Then, and having all these different variety of structures, you can use them. No, you can do some whenever you think that docking experiment with different structures. We had lastly published an article where it opens and then large structure can dock inside. If not, it cannot dock. Then you have all this variety. You can do virtual screening on that. You can do docking with proteins. When one is very flexible, it has to open in order to dock the other one. And you can think many, many different things. And then which is very difficult to do only with dynamics. Okay, then then you can have many questions to ask. Then what are the sufficient number of normal modes to consider? You will not be taking the 3n normal modes, okay? Because it's 3,000 directions. You'll take maybe the first 3, 4, or 5, which represent these changes. Then this, you have to choose them also, maybe with other criteria. Then after you have to combine the modes, okay? Here you have to combine, you have these different vectors. How many combinations that you will be doing? It depends on the number of modes. For example, Antonio Gomez, who was here, he showed that if you have 3 normal modes, you must have minimally 12 combinations. If you have 4 normal modes, you have 24 combinations, etc. And also, what are the sufficient number of kinetic excitations? How many excitations you have to do here? 10, 20. If you do more, you can dislocate your structure. And how much kinetic energy you have to put? Not very much. If you put very much, then I will push away, and you'll be dismantling your structure. And then you have to also very adiabatic, small pushes. Then these are many questions that we ask to ourselves before starting the simulations. Then Mauricio Costa wrote a new some algorithm, some program which he called MD Exciter, which is an R program, and which does all the tasks. But is very easy to use. He reads the file. All this management is done by the R, reading files, all the required options for calculating the kinetic energy, etc. He compute also the normal modes using the Bio3D, but he can read externally some normal modes too, or PCA, anything. And then he adapts the inputs for the different programs like NAMD, CHARM. I think Mauricio will be extending this to other programs. Then it's very convivial, very easy to use. I was here 3 years ago. We were using only CHARM. It was very difficult with CHARM because people are not use so much CHARM now. And in AMD, you have all this GPU, which is very rapid, fast. And then this friendly use of this MDNM procedure. And he does also many re-excitation too here. Okay, I think it will be shortly available in GitHub. Yeah. And then for the normal mode computation, then initially we have been developed by using physical force field, as I told you. But for large system is very costly because you have very big matrices, because 3,000 columns correspond to 1,000 atoms, which is a very small protein. And then, but now you have big systems, and is not possible to compute the eigenvectors and eigenvalues. Then the second way to do is use elastic network models, where you have some springs between some atoms of the protein. For example, it could be only the C alpha atoms, which is called elastic network model. Then you have all the springs are in rest. Then you don't have to minimize. He's already in the minimum. Then you can have different selection of atoms. Then disadvantage is that the matrix is smaller and you don't have to do energy minimization. And another method is rotation translation block, where in each block you can put a whole residue, like a rigid. You can put 2 residues, 3 residues, or 10 residues, depending on the size of the protein. And then you have a limited number of degrees of freedom, because each box has six degrees of freedom, 3 translation, 3 rotation. And then you can consider all the atoms. You can consider the physical force field, but you can also consider the elastic network model. And then you don't have the energy minimization. It depends if you use ENM. And this is also widely used, which is very interesting. But you don't have the side chain flexibility of the residue because it is in the blocks, rigid block. But you have his displacement of the residue. Okay, then, and the frequencies are good if you use the physical force field. It's just the shifting. The frequencies are shifted to higher frequencies because of the rigidity. But it's just translational. You can recover the frequencies if you use the physical force field. But this is not the case with ENM, with elastic network model. Here's the frequencies. One corresponds the histogram of the frequencies. The black curve corresponds to the physical force field, is the amber force field. And the dashed ones are the simplified elastic network model. You see that is not the same histogram. Then you don't have the same distribution of the frequencies. And you know that frequencies are very important to compute the thermodynamics quantities. Then some has to be very careful considering the frequencies, which method they use, okay? But I will not present here the thermodynamic quantities. Okay, yes. Which block?
This method of blocks, what's the difference from the coarse grain block?
Here you don't have blocks. Each residue has, you have taken one point, one atom. In the block you have many atoms. If you have a residue, you have the whole atoms of the residues. If you have 2 residues, you have all the atoms. And then when you do the gradients of the potential, you take all the interactions of all the atoms, which is not the case if you take only one atom. You can use physical force field in that case, in this box, but you cannot use physical force field in this case. Because you don't have, between C alpha, you don't have a cohesive force, no? You don't have physical, is very, you have put a spring which should not exist. But it's not the case with the block edit. And all the interactions are taken into account. Okay. Yes? Yeah? Okay. Offer is a projection in the space of all these vectors. It's like you project, if you have a surface, you have a vector, you project on a given coordinates, x or y. You have diminished the number. Here also you diminish, you have the whole Hessian matrix, which you project on a more limited number of degrees of freedom. It's like a projection. You change your, also in quantum chemistry, they do also this kind of thing, matrix projection in order to decrease the matrix size. And then you have smaller matrices, then you can compute. But you have all this interaction between the, yeah.
Because in the RGB, the blocks, in the normal modes, with a folded protein, there are a lot of positive motions where there are a kind of a local rigid block mode that moves together.
Yes.
So you can do this because of the characteristics of the movement that some domains of the protein are moving together. So it works because of it, no?
Yeah, maybe I didn't understand very well your question, but you obtain the same if you have domain motion. You'll have the domain motion.
No, because for example, you have two domains.
Yeah.
Within the domain, this domain, for the motions, this domain moves like a rigid body.
No, but the domains, if you have many blocks in the domain, then they will move. Also, you have internal domain, internal motion within the domain, but you have also the lowest frequency will give you the hinge bending motion. And I don't know if I answered this correctly. You? No? Okay. It doesn't work. This is just a comparison. That is an article which was submitted with, which was very interesting article, comparing the physical force field for a given protein, the projection of conformation generated by this with MDNM method for a given protein, I think is lysozyme. And this, using only physical force field, this all atoms elastic network, this only, this RTB, is, I think is all atom, is RTB. This only the C alpha atoms. This is the PCA, because this, you can have thousands of structures or given experimental structure. You can do the PCA of this structure. And then you can use MDNM to have the exploration. Then we compared all these methods. You see that the MD explores very tiny region. With MDNM, you have larger region explored. And the other methods are very comparable. Then it's justified that using only C alpha atoms instead of using physical force fields. But the simulation is done with physical force field in MDNM, use physical force field for simulation, but use only the direction of motion. Then is a good strategy using ENM, elastic network modes, rather than all atoms mode, which is more quicker and more rapid to explore the space, the conformational space. Okay. This another problem with normal mode is that if you have a closed structure, you have springs in between, then you cannot open because it's glued here. But if it is open, you can go to the closed form. But this is also valid not only for elastic network model, but physical force field. If you have very closed cohesive structure, it's very difficult to open. This is a real problem. And then we tried, I have a student, then we were very annoyed because he had to analyze the motion of this transport protein, which is ABCG2 proteins, which is a membrane protein. This is the internal of the cell, this is the exterior. Then it expels the compounds which are in the internal. It has to go through this ABCG2 to outside. But it has, first it opens here, this open outside facing state is open here. The substance come here, but is blocked. Then it has to change, and in order that this is a change, then and the other part opens that the substance can go out. And we wanted to study how is done this transition. And it was not possible with normal modes because it was very closed. You cannot open it. It's a very problem. But we were able to do from the other side. If you go from this to this, it's more easy. Then, but we wanted to open it anyway. Then this student, which is Balin, who is doing his PhD, he said he had the idea, well, why not to take the direction which points from this, for example, from the closed to open state. If you have the experimental, we have the experimental structure of both of this structure. Why not, instead of taking the normal mode direction, take the direction which points to other, like morphing, you know. But using the MDNM, just exciting and push it away. And we did this. Wow, what's this? It doesn't work. Oh. Then then we tried this transition. We pushed the system, taking this vector like a excitation vector. Then we push the system with kinetic energy, but every time he reoriented this vector to the target, which is somewhere here. But you have to know the target structure. If you don't know the target structures, you cannot do this. Then he called this kinetically excited targeted molecular dynamic simulation using MDNM. But this also comes from an idea of Mauricio, which we used this for computing the internal normal modes during the simulation, recompute the normal modes, and push the system, for example, for electron density maps fitting. You can recompute the modes. But here I don't know, after having pushed away, we didn't try that to recompute the normal mode. Maybe you'll have some normal modes going there. Then this is the directionality problem. To follow a single initial direction might not be sufficient because during the motion, you can have important deviation. If I go there, maybe I will have to go from there. Then I don't have this direction initially. And then if you have large rotation, if I have to rotate my hand like that, if I have a mode going there, but I will not be going like that because it's orthogonal. And then you can have large rotation, which MDNM will not be sufficient. You can have complex motions. And then the second idea is to recompute the normal mode, as has been done by Mauricio Costa, which we have the MDNM EM fit, electron microscopy fitting. You recompute the normal mode at every step in order to direct to the target. But you have to know also the target structure. And then what we developed lastly is just why not to continuously modify the mode direction. Although that I am going, then why not, I know what will be during the dynamics, what will be the forces coming from the environment or other thing, then I can change continuously my directions and then adapt. Then we call this continuously modifying this adaptative MDNM, which the idea is like that. You have the initial direction, then this is the normal mode direction in yellow, and then when you are pushing, maybe your effective motion is going in the black one, not in the yellow one. Then after a short simulation, we analyze what are the angular deviation. If the angular deviation is small, we keep the direction. If the angular deviation is big, we take, for example, here the deviation is big, then we took this one as a new direction, excitation direction. And then we do this many times during the dynamics. And then you are changing the directions of the normal modes. And it's very adaptative. And then we are writing the article at this moment. Then just to show you for the calmodulin. Calmodulin is a very flexible protein. And then you have two domains, which is calcium binding domains. You have a long helix here. But this then is implied in many biochemical processes. And it can bind many different proteins. He can fold, have elongated structure or curved structure. And then if we do, for example, we took two variables here. One is the distance between the two center of mass. If it folds, this distance will be shorter, okay? Because the two domains come together. And then you have also rotation of these two domains with respect to the alpha helix here. You have a linear alpha helix, which we put a pseudo-diagonal angle here. This is the two center of mass, the ending points of then you can compute the phi angle of this pseudo-diagonal angle. And then we did these MDNM simulations and also free dynamics and projected here in abscissa is the end-to-end distance. At the left is our very folded structure, and the right is open structure. And then we took a starting structure at very open structure, elongated structure. Then if you do a free dynamics, I don't know if there were here 200 nanoseconds. And you cannot explore, the exploration is very limited. Even you spend a lot of time, you don't explore. Even not and all these red points are the experimental points. There are many points. But you explore very tiny region of, this is torsion again. In MDNM, you have only the open structure. You don't have the folded structure here. It's very difficult. Because why? Because it's rectilinear direction, it cannot rotate, or maybe you have other internal strains also. Yes?
What is the red points?
Red points are experimental points, which projected there. Okay. Then all this folded structure is not explored. And if we take the adapted MDNM, as I explained you, we cover all this region because the direction can have the possibility to close because he changed the direction. Yeah. Yes, we checked. I'm not presenting here. Everything is preserved, and we checked. We computed the energies of each structure, which is okay, and then everything is well checked. And
the experimental comes from crystallography. the experiments comes from? What method?
Ideas mostly are, is crystallographic mostly, Maybe there are some NMR, I don't know. Okay, then you see how powerful could be this adaptative normal mode. And then we also, another interesting thing that we did is just why not, for example, we have the directions of motions, excitation motion. We projected all the conformation during the dynamics to these directions. You have the fluctuation along the pathway. You have the pathway. You project all this conformation along the pathway, and you can do an FFT, a fast Fourier transform, to see the frequencies, how it evolves during the evolution. And then it was very interesting to see this. These are what we call the spectrograms, just fast Fourier transform of fluctuation along the pathway. And then this is MDNM, and this adaptive MDNM. And then we computed also the energy of, it goes from 0 to 35 centimeter minus 1. In black side, the stored energy, vibrational stored energy, which is omega squared times the amplitude square, is the sum over all the frequencies that you have here. And more it is, then is the black curve here. And when it is very red, it corresponds to quite more energies in the vibration, the overall vibration up to 35 centimeter minus 1. Then what you see is that the energies are much smaller with an AMDNM. Now the values are smaller than MDNM, which means that the MDNM shows a more relaxed pathway than MDNM because it changed the directions, and the stored energy is much less. These high frequencies are less required to change these directions.
Is this for other protein? Is this for other protein?
Yes, this is another protein. But this is a T4 lysosome, just to see the effect. The T4 lysosome, you have the rotation of these two domains, but after it comes together like that, then it will shift to another direction. But in MDNM, if you took only the lowest frequency, then you will be blocked. You will not doing the other direction, okay? And then, this one thing, and you see that there are more high frequencies excitation. We have more red bands here than here. And the only red bands here, the very intense excitation comes when you have some important changes. If you have two modes coming like that. And then when he wants to change, he excites all the other high frequencies, which helps the change of the motion. Okay, and here is something different because you are pushing the system. You must have some barriers. You have to go up the barriers, and it requires also some excitation of higher frequencies. But here is more relaxed, except when he wants to change the direction of motion. Then here the two conclusions of this is MDNM shows a more relaxed pathway than MDNM. And the other fact is important conformational changes occur with excitation of higher frequencies. Then higher frequencies are needed to be able to change in the low frequencies. I told you that there are some couplings, when you go beyond the harmonic approximation. We have couplings between normal modes. And here's an example how the modes can be coupled to help doing things, okay? And then we are trying to finalize this. Okay, then I will maybe finishing here. Some acknowledgement, Brazil group. Then Tulio developed this adaptive normal modes with Costa. Batista was the original MDNM development. It was in it was in. We are continuing to still to develop, and in France there are many groups which also implied. Then, thank you very much. Then maybe I hope you ask me questions. Maybe it was too much fast because I tried to do a compact presentation, and maybe normal mode is also a difficult concept for beginners or doing stuff. Is maybe complex to understand everything on the normal modes. But okay, then.
Thank you, Professor. Now I will open for questions. Yes, you can thank him. Yes, thank you.
But don't be shy to.
Yes, please fire your questions. Just wait for the microphone because we have people listening to us online.
And then I'll become the presentation DJ in there to help you, the guys. So in adaptive MDNM, how do you choose the direction to which you are applying to the normal modes or how do you evaluate if it's a good direction or a bad direction?
No, we don't have good direction or bad direction.
Yeah, I mean, it's not a good or bad, but a plausible or not plausible.
We do that test. We took, for example, the lowest frequency. We excited. We try to see what is happening, how it's changing. We know that the molecule has to close then, and we see that the lowest frequency close the structure. Then we try to see whether it will do the other motion, and we see that with adaptively is being able to change. Then after we took 2 modes, 7 and 8, we excited both of them, all the combination to see what's happening. And we see that mode 9, 10, 11, 12 are being excited too. And we did many tests like that. Then these are many questions that people ask, how do you choose the normal modes? But if you have an idea of the target, then you can choose accordingly the normal mode. If not, you have other criteria.
If you cannot operate on a native fashion using this, because you must know what are the conformations you are trying to achieve, right?
No, a priori you don't know. Then you're exploring where you might go, which could be interesting. Yeah.
You cannot use some kind of gradient to follow the direction or to look for energy or something like that. It's just
But the gadget is, but if you have a target, you can devise. You go to a given direction, but which kind of targets you want?
I don't know. Maybe you want to stay in the valley.
Yes.
I don't know.
This could be interesting areas of exploration to devise new methods.
So we can continue our collaboration.
Yes, sure.
Thank you for your presentation. I was wondering about the difference. For example, you were talking about because you are exploring the normal modes and where the protein goes. Because I was thinking, I kept thinking about stirred molecular dynamics when you were presenting the adaptive, excited molecular dynamics. I thought about the stirred molecular dynamics because but the difference, I think, it's you know where to go, where the stirred.
In the stirred, you know. But here you don't have any idea where you go.
Okay, that was I was wondering the difference. Thank you.
Okay, one more here.
have a curiosity. Can you employ a normal modes for a complex protein RNA, protein DNA? It's like if you have a cryo-EM, for example, 2 cryo-EM, you have a complex DNA RNA. Can you employ this to such a complex?
We are now starting to study the nucleosome, which is, you have histone core, 8 histone core, which is wrapped by DNA. Then there's a lot of question now is very being, there are many research going on nucleosome, how the DNA is expressed, and which this nucleosome has to open. And then in order to study how this nucleosome unwraps or wraps around the histone, there is nucleoprotein complexes. Then we are now starting to apply this MDNM to study this type of motions, unwrapping motions, and yeah, we are starting. And then maybe, yeah.
Okay, any more questions? Okay, I have one. So, how realistic are the structures that come directly from normal mode analysis? Can you actually use them right away, or do you have to prepare them in some fashion before you can, for example, dock some molecules onto these structures?
Okay, but there's nothing to do with normal modes. It's a normal mode is so tiny motion that is around your structure. You have always a good structure. Then is one, you go beyond this approximation very far, then you have to be careful. And then is one of the questions of Paulo Ricardo. So if we have analyzed the secondary structure, then we always do now to see whether your structure kept his identity or not. And then some has to be very careful when it goes very far away. Yeah, to be.
Okay. Okay, so let's thank.
You have.
Yeah, do you have another one? Sorry.
Are you? No,
Oh,
okay.
Okay.
No, just to say that the collaboration with Brazil is from this cooperation between Brazil and France.
Yes,
Cups,
I
Coffee
didn't
Cube,
say.
you have not thanks that.
Yes, it comes.
Yeah, this comes from this collaboration that we have.
Yeah, I put all the Brazil.
Coffee Cube.
Huh?
Cups, Coffee Cube.
Capes Coffee Cube, yeah. But we did more than Capes Coffee Cube. No, no. I came here in 98, no? But we started, but there was no Capes Coffee Cube. But then after there was a Capes Coffee Cube twice, no? And then there are many outside collaborations too, I think. Yeah.
Okay, so now shall we thank Professor Perahia one more time, and please give him his salute.
Thank you for listening to me. Maybe it was difficult in some places.
Well, it is really hard, even for those who work with that for a long time. So,
yeah, I recognize this because when I started, for me it was difficult when I started.
Amazing. So thank you, Professor. Thank you, everyone, for coming. And yeah, until next Wednesday.
Yes.